So I'm a basic scientist working to try and understand what's going wrong in Parkinson's disease so we can then identify how to correct that with therapies, which then feeds into why we would test those in clinical trials to see whether our analysis was correct or not. So this is not meant to be a lot of detail on this slide. Let me just turn the volume up, I think. What we have here is over time, and at this point here, 0 at time of diagnosis, and really to get across that there are a number of symptoms, not just motor, but also non-motor, and that some of these symptoms are appearing prior to diagnosis of Parkinson's disease. So this is an assembly. It's not meant to be a rigid template of what's happening, but here is basically with time, prior to diagnosis, there are some symptoms appearing. And then at the time of diagnosis, you're seeing the appearance, obviously, of those motor symptoms, perhaps new non-motor symptoms that are worsening. And really, this is to get across the disease progression, that basically over time, this disease is progression. And as Michael nicely conveyed to you, that we have treatments that are helping for those symptoms, but we don't have any diseases that we have validated, any treatments that we've validated that actually prevent the progression or slow or stop it. So my lab has, in terms of a Parkinson's aspect, has two main themes. And the first one is to try and identify the early events in Parkinson's disease so we can see if we can target those problems with what's called disease-modifying therapies, which would slow progression or stop it. We would be happy to start with slowing, and we'd love to then fine-tune that to stopping. But there are aims as to find out what is the problem, what's going wrong, how can we fix it. The second aspect, which goes hand in glove or dovetails with this approach, is to identify biomarkers. So you may be familiar with biomarkers if you've ever had a resting blood glucose test. You give a sample of blood, they measure your particular modification to your hemoglobin, and that shows how well your body's maintained or controlled its blood glucose level, which is an indicator of what could be a precursor to type 2 diabetes. So it's a simple blood test. You get a number. 5 is good. 6 is a little worrying. 7's not good news. But it's a simple test. There's a biomarker that's giving you an indicator to the clinician what's happening in your body. We urgently seek those in Parkinson's disease. So not just looking at your symptoms, could we have a biomarker measurement that's going to be quite quantitative, and that would give us an indication of, does someone have Parkinson's disease? What stage of the disease they're at? They have a lot of benefits for us, though. They might help us diagnose patients before the Parkinson's. So if here you can see if this is the course of the disease, we're diagnosing relatively late in the disease course. It'd be much better if we could diagnose much earlier. We also, when doing clinical tests, what's called drug efficacy tests, asking, are they effective in stopping progression, we are using whether the symptoms get worse or not. Biomarkers may be far more quantitative and help us to really define and clearly indicate whether those drugs are working. Not just as a group of a population of people in the clinical trial, but also as individuals, a so-called precision medicine. A drug that works on 80% of people may not work on 20%. We could monitor you through those biomarkers to see, is this drug working for you? If not, could we switch you to another one? Or it is working for you. Let's keep you on that. So there's a number of very important uses for biomarkers as well as therapies. So in that earlier slide, my first aim of my lab is looking at, can we identify these early events in the disease? And why are we interested in that? Well, hopefully it's clear, but sometimes things I talk about them so much they're clear to me, and I forget that perhaps not everyone's in that field. So if you consider this disease as a problem, it causes more problems, and it's a cascading of events, as I've tried to convey here in this cartoon, or a series of dominoes that knocks over one and then the other and the next one. We're best to basically target to intervene for therapy. So why target the early events? Well, let's take the flip of approach. What if we target the late events? So down here, we've got this cascade of events, and maybe just over here, we say, well, let's target that event, that particular symptom, that particular defect in the brain region. Let's target just that specifically. The problem with that is that even if we're successful and adjusting that with therapy, then best case, if we have a therapy shown in green here that stops that progression, is we end up solving that symptom for a patient, but all these others still remain. So far better and smart is to look for these earlier events that are happening upstream or in those events in different brain regions, because if we can target those, none of these events take place if we're successful. So that's why we're looking for early events. So let's say we find that. We have a therapy. We could give those to existing patients. So these are people who are post-diagnosis. We give them their therapy, and if our therapy is very successful and actually can stop progression, then my clear sign here of the stop sign shows you that, okay, that would, in a best case scenario, stop you progressing so your existing symptoms don't worsen and you don't develop additional symptoms. So that's great. But wait, there's more. Could we give that, what happens if we give that therapy to someone who's just been diagnosed? Then, compared to the last slide, we would freeze, no pun intended, or stop people right at that stage. So their symptoms would not progress and worsen. Well, why stop there? Why not see if we could give that therapy to people before they show symptoms, so that they may show these smaller symptoms, they're unaware that they were precursors to Parkinson's disease, and just to make sure none of these specific symptoms that tell you might have Parkinson's disease, so please don't panic at this stage. And so we're adding therapy. If we could identify patients much earlier, before they show clinical symptoms, that's our goal in the biomarker field, to try and be able to identify people much earlier. Because if we stop you there, you never have these symptoms. You know, it's effectively a cure. That's what we aim for. But it's a hand-in-glove approach that we need to have therapies that stop disease progression, as well as being able to identify you much earlier in the disease course so we know who to give that therapy to. So how's this magic trick done of identifying people before they show symptoms? This is a completely hypothetical cartoon, which is what I want to get across the point of why this has benefit. So age is the biggest risk factor for Parkinson's disease. So as we age, even in the normal brain, and we're just showing a hypothetical situation, an individual who's not destined to get Parkinson's disease might have a slow decline with aging. And although this is just a cartoon, I've done an abrupt thing where some person who's going to develop Parkinson's has a much stronger slope or loss of neuronal function, and eventually that will cross some threshold and they'll start to present with symptoms. Maybe they're motor symptoms because they have insufficient neurons in that specific part of the brain to perform all those functions. So they now have things like those clinical symptoms associated with that. So if instead this is where the neuronal loss starts, a time later, this is all hypothetical please, there's our diagnosis, then this is, as I've shown representing before, this sort of pre-clinical symptomatic stage. If we could instead identify these people here and detect them much earlier, and in this case I'm just slowing neuronal loss, I'm not stopping it, then you can see that slope is much longer, much shallower. And the distance from when they would have reached the threshold stage of age of standard diagnosis to over now, that's the time we've given the patient before they start to show those motor symptoms. In my little diagram here, that could be 15 years. But this is what I'm trying to get across is why, if we could identify patients early and have a disease-modifying therapy, how coupled together they really offer great potential. So, as I said, how do I do this magic trick of identifying people before they show symptoms? This is what are called biomarkers. We're looking for things that would indicate the status of your disease so that we can monitor them, and they change with time. Now, there are different, these are in a hypothetical situation, there may be something that is changing and we can measure early on in the disease course that would help us with diagnosis. There may be other biomarkers that are changing with time later on in the disease course. They may reflect changes of cognitive impairment, or they have your motor symptoms. We have the possibility to look and hopefully identify different markers at different stages of the disease, because they all would be different, very useful for different aspects. So these would need to be, biomarkers can be taken from different places. We've chosen to look for them in blood because it's far less invasive. Obviously, no one's going to give us a frequent brain sample to check whether you have a problem. So there's a lot of consumer pushback there. And so that's why we're looking for what are called peripheral or blood-based biomarkers. So here's basically what we're looking for is understand the disease mechanism, because that'll help us identify therapies, but in addition, having those biomarkers acting in this trifecta to help us do that. Right, so the early events, how are we looking to identify some of those early events? Well, it's an endeavor, as Michael's touched on, because in people who have idiopathic or sporadic Parkinson's, we really have a feeling that it is genetics and a combination with interlacing with the environment, but we're still searching for what are those roles. So to have a therapy, we have to understand what's gone wrong. So that's why we're looking for what has gone wrong. And this is just that slide to remind you of where it was before. So as Michael nicely touched on for me, rather than is it genes or is it environment, as one can imagine these complex diseases, it's an intersection of both. You have a loaded gun here, and then is this in some way some exposure, whether you're exposed to pesticides at some stage or those type of aspects, are they playing a role as to whether you end up developing Parkinson's disease? So this is to show genetics that we do a lot of here at the Garvan, and environmental impacts. Some are protective, so drinking coffee is good, but damaging or increased risk could be pesticide exposure or brain injury. They are increasing your risk of Parkinson's. And both of these are interacting in some way to have an outcome as to whether Parkinson's disease eventuates or not. The approach we've taken to find out what are those early events going on in the human brain is to look at what is the output of your DNA as influenced by the environment. So just to give you a small refresher course, here is DNA that's encoding all your genetic information. It sits in a protected environment in your cells, and when the instructions or blueprints are there, it makes a temporary photocopy of it called RNA, and how much that RNA then is then translated into a particular protein or enzyme that carries out function X, Y, or Z in your brain. So DNA makes RNA, makes protein. How much? Whether you have too much of a protein, whether you have a protein that's not working, you have too little. These are the indications we're asking about what's different between people's brains with Parkinson's and those who are healthy individuals of a similar age, and that is to look at, we measure the RNA levels of every gene. So the Garvan's genomic facilities here allow us to take samples of people who have generously donated their brains to science. We take the RNA from those relevant brain regions and do a lot of sequencing without going into detail that's unnecessary here. We're trying to identify what genes are inappropriately on too much or too little, and therefore that would tell us what cellular pathways, because I'm a cell biologist, really what are some of the pathways that are disturbed in patients. Many of those pathways are normal between healthy individuals and patients, some are not. If the pathway is not working, can we somehow enhance it? If a pathway is overworking, can we inhibit it and dial it back? So they're the approaches we're taking, and it's a lot of data analysis. Whoops, I'll go back one slide. It's a lot of data analysis. We're following up on a number of those aspects and with the pathways to see whether does that mesh with other people's thinkings, are there drugs that are already available that we could repurpose that are therapeutic in other diseases, do they share a target, and could we use those for potentially putting forth into trials for Parkinson's. So that's ongoing work. One of the outcomes from this endeavor is, as I said, Parkinson's disease is a progressive disease, as Michael also mentioned, and this is showing with time, this protein that he also mentioned of alpha-synuclein, it forms these lumps or aggregates, some of you may be available, called Lewy bodies, and German Brack analysis who looked at showing Lewy body patterning is in essence saying that with time, you see more and more of these Lewy bodies, an indication of pathology in the brain as the disease progresses. It's always been a very intriguing question for scientists and clinicians as to what's underpinning this progression of more and more brain regions showing pathology and damaging. So we're very interested in that, and then a very intriguing theory or hypothesis came up a number of years ago that alpha-synuclein, this protein that we associate with the disease, and we know that in cell culture and experiments can be quite toxic, that it may be transmitted or transferred from one cell to another. And that, in essence, I'm using my word carefully here, it is spreading from some of these neurons that are affected in this brain region, and with time, spreading to other interconnected brain regions. That was a very intellectually appealing argument to explain perhaps why we're getting this disease progression and accompanied symptoms. Now, when I talk about spreading, what I really want to point across here is that this is not an infectious disease. We're talking about spreading within the brain. not to your relatives that you're talking to. So just to make sure we don't go away with that thought. As a cartoon, this is a prion-like spread where here is alpha-synuclein, can be in two forms. In green, it is in a manner that is doing its normal function and working appropriately. But if it changes its shape, or what's called misfolds, as shown in red, this neuron might have too much alpha-synuclein that's toxic. It's impairing the function of this neuron and contributing to your symptoms, and that in some way, in a non-deliberate manner, this toxic protein is released and regretfully taken up by a neighboring neuron or brain cell, where it can coerce these normal green alpha-synuclein to take on this altered shape, and you start to see this transition, and before long, the second neuron is now full of the toxic species. This is our hypothesis, and we're having this individual neuron is no longer working, and it's continuing on. So it's this transmission from cell to cell of toxic forms of alpha-synuclein is gaining a lot of weight as to what's perhaps underlying the spread of pathology in Parkinson's brains. So obviously we would love to be looking for therapies that could stop this transmission of cell to cell. If you could interfere with that, then that would be another way of stopping disease progression. So depending on where we gave it, that's what we're searching for. So I'll just touch on one thing of a gene we've worked on called Park9. It's a Parkinson's disease. Rare individuals have a mutation in it and end up with Parkinson's disease or Parkinson's-like symptoms. If you lose Park9 in our mouse models, you get a lot more pathology, suggesting maybe it is spreading faster. That's one approach. We didn't want to find a compound that accelerated the problem. We were looking for the opposite of that, obviously. And so we decided if we could overexpress Park9 and make more of it, maybe it would slow the spreading of this. So we're quite excited with Louise Cottle in the lab, has recently taken mice that we force or genetically require them to make additional levels of synuclein. So they're a model of Parkinson's disease, and they show some symptoms. And then if we put a virus into their brain that forces them to make more Park9, we were very excited to see that. But in doing that, that could reduce some of those Parkinson's symptoms. So that's early days, it's in mice, but it's very encouraging for us to want to continue working on how we could elevate levels of Park9 as a potential therapy. So in the time remaining, I'll just touch on the PD biomarkers front. That's that second component. If us or others have therapies for testing, we need to be able to identify people earlier on to give those to them. So we're looking for PD biomarkers. Again, we've chosen to look in the blood of people because it's easier to access rather than a CSF sample or something like that, or more difficult samples together. And all of the RNA changes that we identified in the brains of Parkinson's patients, that gave us a huge data set to then say, well, if these are changing the brain, we hope that if we look in people's blood, do we see some or all of those same changes? And so we looked at depth at a number of samples. It was a Michael J. Fox supported project, and Shake It Up Australia. So thank you for that, Clyde. And what it was to take blood samples from patients and controls and then see if we could find, are there particular patterns of these RNA molecules in the blood of patients that we could distinguish patients from healthy individuals. So that was, there's not going to be one single molecule, I don't believe, in Parkinson's that's going to be the perfect biomarker. So we searched for combinations of RNA molecules that had changed in the patient's blood to give us an indication of, could we identify in a blind manner these patients who we knew had Parkinson's, but we were blinded by that, and identify who had Parkinson's and who didn't. We were very accurate with that. We were pretty excited about that. Now, I've got to say, on the first set of people, we could do that very nicely. The real crunch is, can we do that on a second set, what's called validation. So we're underway to do that, to test and really show that these biomarkers would work, and I hope I've conveyed to you how the benefit of that. So with that in mind, we've led a Garvan-led Australian-wide initiative to bring clinical trials to Australia, as I spoke at the introduction, and that's why we've reached out to members that you are hearing from today from the Cure Parkinson's Trust and Link Clinical Trials, because part of it would be that the biomarkers would be useful for all of our clinical trials. If they are working, they will help us, as I said, test whether those drugs are successful or not. And that's why we're forming this partnership. As I need to say again, we're in the planning stage. We don't have drugs for you to contact us about yet, but once we do, we will be making that known. And finally, I think that's where I'm going to finish up, to just say the importance of this sort of combination And then we've got a 1-minute movie from Tom Isaacs, who's president of the Cure Parkinson's Trust UK. And I believe we hopefully will be able to show that. And then Richard will be talking.
Hello, my name is Tom Isaacs, and I'm president and co-founder of the Cure Parkinson's Trust. I've also been living with the condition for over 22 years now. One of the most exciting and innovative projects I've been involved with at the Trust is our linked clinical trials program. Nothing like this exists anywhere else, and nothing else has the potential to deliver treatments with the capacity to slow, stop, or even reverse Parkinson's in such a short space of time. The initiative brings together some of the most eminent Parkinson's scientists from around the world to prioritize from a fully referenced list of drugs already approved for use in other conditions such as diabetes, heart disease, cancer and cystic fibrosis, and all of which have strong biochemical reasons which suggest that they also could greatly improve the treatment of Parkinson's. Linked clinical trials responds directly to the sense of urgency experienced by all of us who live with Parkinson's. And it is hugely encouraging that as well as enlisting support from the States, in particular the Van Andel Institute of Michigan, we are now also generating significant interest and backing from the Garvan Institute in Sydney, Australia. There is one thing that is more likely to provide a breakthrough and ultimately a cure for Parkinson's. It is teamwork. And the Cure Parkinson's Trust is acting as the glue, bringing everyone together to concentrate on what's really important in Parkinson's.
All right. So you can see our excitement here at Garvan to bring clinical trials to Australia as part of our endeavour with Parkinson's disease. And to talk our last talk for today from Richard Weiss, who's Research Director of the Cure Parkinson's Trust and works tirelessly to identify drugs and bring them into clinical trials. And he'll tell you what it might mean for Australia. So thank you.